I'm Jonah Comstock, Editor-in-Chief of pharmaphorum. I'm here at JP Morgan, actually at the Biotech Showcase at the Hilton in San Francisco. It's Wednesday. I'm joined by Michael Bauer, the CEO of Quantro Therapeutics?
Quantro Therapeutics in Vienna, that's right, yes. Austria.
Oh, great. Long flight for you.
Yes, long but pleasant, no problem.
So tell me a little bit about Quantro. What do you guys do and what sets you apart?
Yeah, so Quantro is dedicated to make the field of gene transcription factor control accessible. So we're actually the only company that can measure the transcriptional dynamics in a living cell in response to a therapeutic intervention. So and that sets us apart from older methods like RNA-seq or next generation sequencing approaches, which are defining steady state at a certain point in time, but the technology is not suitable to measure immediate response on transcription. So if you imagine you take a drug, your body responds within minutes. RNA-seq can only see something changed after 12 hours or longer. Now by that time, a whole cascade of things have happened. So you will never be able to pick up an immediate instant response, and therefore also not understand why you end up at the new steady state. The other issue is RNA-seq only quantifies RNA without any understanding on the dynamic. Like if you have a given amount of RNA, you have no idea on the turnover. That's a separate study, and that we can all do now in one shot. And we can measure changes in transcription as early as 1 hour after treatment.
Wow.
Now, this is completely new, and we're the only company that can do that. And even more so, we have translated this into a high throughput screening system, which allows us to screen hundreds of thousands of compounds for new modes of action drugs for currently non-druggable targets. So the issue that's shown in this headline, if you like, the issue with drugging the so-called undruggable targets like transcription factors has not so much been that they are not druggable, it has simply been not possible to make meaningful assays. Like, for a transcription factor, you can't take them out of the cell into an in vitro system. Yeah, they're too complex. There's multi-protein interactions with DNA in a living cell. You cannot reconstruct this in a cell-free enzyme assay. This is too complex. You can also not use reporter gene assays or green fluorescence protein type approaches because again, the time to get a signal is so long that you're not picking up the primary response. So this is unique to what we do, and for this we open the door wide to any transcription factor in any indication because the technology is universally applicable to all indications. Now, we are focused on cancer. Our lead program is CMIC, again, the famous non-druggable, intrinsically disordered, multi-protein complex. Many people have tried to find something on it. And again, there's not really a suitable assay for doing that. You can do binding studies, and a lot of stuff binds to MYC, but you have no idea if the binding event is actually meaningful. So a lot of companies that do chemoproteomics and claim to have a drug binding to a target, that's not wrong. But the question remains open, does the binding have any effect?
Yeah.
And even if you measure an effect, is what you measure actually true? Because if you're using next generation sequencing, a day after an intervention, many drugs appear to be a MYC inhibitor, but they're not. If you take a KRAS inhibitor, you wait a day, it looks like a MYC inhibitor because MYC is gone. If you take a Notch inhibitor, you wait a day and MYC is gone. It's nothing to do with hitting the target directly. And the issue with targeting MYC is upstream, it works. There's more than a dozen pathways feeding into CMYC to promote cancer. So if a cancer cell has this pathway living on MYC, they have a multitude of options to get MYC activated. So if you block one pathway, let's say bromodomain, that works for a while, patients relapse because the moment you block bromodomain, for example, Notch is upregulated. So there's an alternative pathway. So that's why we want to target MYC directly, and we're actually now succeeding. We have now a series of hits from different screens for different modalities, small molecule, lead-like compounds, fragments. We also do some rational design approaches, So we now have drug candidates emerging that are directly interfering with MYC, and we see this as a functional readout. So how do we get to know the functional readout? That's the core of our approach, is that we actually genetically engineer the target, okay, to make it susceptible to oxygen-induced degradation. So oxygen is a small molecular glue that leads to ubiquitination of the target. So we genetically engineer the target, make it, and then basically by adding oxygen to the cell, we tell the cell to degrade the target, and this triggers an immediate transcriptional response. Okay, and this response is, to our surprise, very distinct and a very limited number of genes that give the primary effect. Okay, and whatever you see a day after is then secondary, tertiary, and knock-on effects like cascade effects down the road. But we can actually now measure the direct impact on the cell, taking away the major component of the cancer pathway in the cell, and derive this search fingerprint. And this is typically, a dozen or so genes only. And this is our search fingerprint. And we're looking for drugs that delivers the same effect. Okay? So we take a functional transcriptomic approach to the target. And we find drugs that do the same thing or something very similar. And the similarity we can also score with our AI data analysis platform we have also generated in the meantime to match all these screens, all the hits we're finding to the right mechanism of action. So and then to the point of how we actually measure transcription, we're using a technology which is called metabolic labeling. So we add to the cell culture a surrogate building block for RNA, which is built into the RNA as it's produced, and then we take a lysis step and we alkylate this building block, and this building block is then leading to a false or a misread in the reverse transcription to make the cDNA from the RNA to do the sequencing. So with this method, we can quantify at any point in time the ratio of old versus new RNA, and from that we can derive the rate of production, rate of dissipation for each RNA species for each gene in a living cell, like 20,000 genes. And as I said, from our genetic engineering the target and removing or telling the cell to remove the target within 20 or 30 minutes, this typically happens, we get to see the search fingerprint, which we then use in high throughput screenings to identify drugs that deliver the same or very similar effect. So in terms of similarity of hits, the perfect match would be a degrader. So our technology is actually the best available method to find molecular glues and degraders for the target, because that would give the identical effect. If you have off-target effects, we will see this immediately, and of course we can use this information to then optimize these compounds for specificity and potency.
So when you have a platform that has such a kind of foundational difference, right? You're at the very beginning of the process. You're doing things differently than other folks. You're able to see the effect of this transcription, more accurately, more quickly. Why go ahead and use that end to end rather than selling that itself as a platform, or to all the other drug developers might be interested in it?
Yes, so we're not selling a platform. We are looking for strategic partnerships with pharma companies. We have already one ongoing with Boehringer Ingelheim in Germany on selected targets in oncology. But we are open to add more than that. So it's 2 targets we're working on right now, but we are free to add more strategic collaborations.
Right. Once you prove out the concept.
Well, the concept is actually proven. We find drugs. We find drugs for targets that have been not accessible so far. So we know the platform works. We are finding drugs. We are able to optimize these drugs, or these drug candidates, and we're taking these in our own pipeline. We're taking this now from hit to lead towards clinical development. So I'm here to raise money for Series A to take our own pipeline into the clinic in the next 2 to 3 years, and to, of course, further exploit the platform. Now, exploiting the platform beyond oncology, we would prefer to do this in strategic collaborations, because all the in-house knowledge is around oncology. So the whole team is oncology. But as I said, the technology targeting gene transcription is a universal principle of life. And most diseases are actually the outcome of misguided or or dysfunctional gene expression or changes from the healthy status quo. So most diseases are implicated in some way in changes on gene transcription, and we can find drugs that modulate this. And modulate means inhibition, complete inhibition, downregulation, or upregulation or activation. So we can see both. We can see up or down regulation, and therefore we can find activators or inhibitors of certain pathways. And that is something nobody else can do in this scale because, as I said, our focus is for targets that cannot be, mainly transcription factors, for example, that are not suitable for having a simple enzymatic assay ex vivo. You don't need us for kinase. For kinase, we can offer the understanding and the measurement to an unprecedented precision if your drug is really on target. Because the issue with biophysical assay-based screening or discovery is you have your binding affinity and you optimize for the binding affinity, and that's all great. You get nanomolar potent compounds, fantastic. And in phase 2, you find out that you have undesired side effects, which you didn't pick up in the SARA panel. We see from the first primary hit, we see how good your compound is on the target and how much you have off-target functional effects, which is completely neglected so far in the early stage development with, if you're using biophysical assay-based screening. There's no panel out there that can cover all these potential issues, but we see them even at the first primary screening.
How do you keep your, what you have there? How do you keep other people from copying it? Once they see what it can do.
We see a lot of growing interest also with big pharma on that, and also we see some activities around that are kind of borderline critical. So we got the patent for the technology. So this whole system for metabolic labeling and the reverse transcription methylation is covered by a patent which we have licensed for the application in drug discovery. It can be used by academic institutions for research, of course. But if you want to use this in the context of drug discovery, we have a patent on that. And you may think, who cares if Quantro has a patent? But of course, we are financed by Boehringer Ingelheim Venture Fund and Ivotech. So if you try to circumvent our patent, it's not us you fight with. We're a big corporation. So we have the technology IP. We have spent, even though that the founders of the company are still active, two academic professors are still active in the company. They're spending about 20% of their time on the company. So they are our CSOs, if you like. And we have built an enormous amount of know-how. So it took us 5 years almost to build up this platform, to make the science into a fully functional transcriptomic discovery engine, which is now running. We can screen about 100,000 compounds a month, so more than 1 million per year. We can also now, and that was a press release that came out earlier this week, we can now multiplex, so we can actually do 10 targets at the same time in a single screen. So just imagine from a development point of view or from a discovery point of view, you're no longer doing one target, one assay, one screen 1 million compounds and do the whole shit again for another set of targets. We can actually do 10 targets in one shot.
So that's a 10x efficiency multiplier.
Efficiency. And because we are not looking at a distinct binding interaction on a molecule, we take it from a functional perspective. So if we see an inhibition of a function, this could be a drug that directly binds and blocks activity. Let's say for CMIC, as an example, with the same assay, we find degraders of CMIC. We find covalent and non-covalent binders. We find binders that bind somewhere to inhibit MIC-MAX interaction. We find binders that are inhibition of the MIC-MAX complex interacting with DNA. We find drugs that are interacting with other important cofactors. So you see the whole universe around the target. You're not limited to just one interaction. and of course that potentiates the ability and the chances to find something interesting on a given target by orders of magnitude.
So I think I have to wrap us up, but for a kind of final question, what are the upcoming milestones for you? What are you looking at being able to announce or even seeing internally in the next year?
So we are aiming for a Series A financing. That's our VC financing angle to advance our company. We are in a very successful collaboration with Boehringer Ingelheim, and we hope to continue and expand that eventually. I'm talking to another pharma companies for strategic partnering on oncology or other indications. So I'm pretty sure we're going to see more positive news during this year. We've had an amazing last year with a number of key milestones, like the first milestone for the BI collaboration, an onward investment of our existing shareholders. We just announced the multiplexing, and there's more to come. Of course.
Awesome. Thank you so much, Michael.
Yeah,
really great hearing about what
Thank
you
you
do.
for your time.
Yeah.
Pleasure meeting you.